Welcome dear students. Today I'll be taking a biology lesson for you. This lesson in biology is in continuation with what you have done in your 8th standard. This lesson is also one of the basic concepts of biology. So let's begin our lesson today.
Let us start our lesson by asking a question. What makes us different from a table or a rock? How are we different from them? Yes, we are living, they are non-living. What makes us different from them is the basic characteristics that we living things have. And on the slide over here, I have a very famous analogy of biology, Mrs. Gren. Mrs. Gren stands for the 7 essential life processes that are found in living things: movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. All these life processes are happening in our body, but which is the basic component of our body which helps us to do all these work, which also will do all these work inside itself. That's the beginning of our chapter. We are going to study the fundamental unit of life, the basic unit of life that every living organism will possess. Yes, the fundamental unit of life is called as the cell. How can we define cell? Cell is defined as the structural and the functional unit of life. Let us compare a layer of cells with that of a brick wall. Now look at this brick wall. The entire wall is made by single bricks put together and cemented together. They are now forming a structure. Together they are forming a wall. In the same way, one by one cells put together forms the structure of any organism. As you're seeing, a peel of onion is nothing but cells that are put together. So, cell is called as the structural unit because body of all the organisms is made up of cells. It is called as the functional unit of life because all the functions of the body, physiological functions, biochemical functions, genetic functions, and everything that takes place in our body is done by our cells. Amazing, isn't it? How were these cells discovered? Let's go back in time. The cells was first described by a person called Robert Hooke, who used a primitive kind of a microscope that you're seeing on the screen right now. And using this microscope, he was the first person to draw the structure of cells. He basically took a piece of cork, dead bark-like structure, and took a section from there and observed it under his microscope. What did he see? He saw empty chambers like structures. And because for him they resembled the cells where monks lived in a monastery, he called them as cells after those monastery cells. Anton van Leeuwenhoek was the person to first observe living cells. And he observed a lot of living cells. He observed the bacterial cells. He observed the protista cells. He observed the sperm cells. He also observed our red blood cells. He observed the nematodes or the worms that we find in our intestine. He observed all these through the primitive microscope that he had created. Then comes Robert Brown, a scientist who was the first person to use the word nucleus of certain structures that are very commonly found in all the cells. He called them as nucleus. Whereas We have a person called Purkinje who is responsible for using the word protoplasm, which is the living thing material, all the living material that we find inside a cell. We have three scientists over here, Schleiden, Schwann and Virchow. According to Schleiden, all the plants that he observed were made up of cell. And according to Schwann, all the animals that he observed were made up of cell. Then comes Rudolf Virchow, who contributed another statement saying that all cells come from pre-existing cells. So together, these 3 points of these scientists were put together to form the cell theory. And what does the cell theory say? That all living things are made up of cells. Cells are the basic unit of structure and function in an organism, and cells are produced from existing cells. Now on this slide you can see, based on the number of cells that you have in your body, organisms can be classified into unicellular as well as multicellular. Based on the size also, we see a lot of variation. We have the smallest of the mycoplasmas to the largest of an ostrich egg. Both of them are single cells. There is also a huge amount of variation that is seen in the shape of the cells. So, cells are diverse based on the functions and the surface area requirements. So, you can see the shape of a red blood cell compared to the cell of a sperm, an ovum cell, a bone cell, a nerve cell. Such a huge variation in cell that is found in everyone's body. Whatever type of a cell it is, it will have three major features. A cell will have a plasma membrane, a cell will have a nucleus, and a cell will have cytoplasm. Let's begin with plasma membrane. Plasma membrane is a double layer structure made up of lipids and proteins. It is, kind of a security guard. It just selects what should come into the plant cell and what should go out of the plant cell. It is therefore called as a selectively permeable structure. It plays other roles also. It keeps our cells intact. It is protective in nature. It regulates the transport of materials into and out of the cell. Materials like oxygen, carbon dioxide, water, etc. can pass easily across the membrane. Now, plasma membrane is what is allowing a cell to exchange its materials with the surroundings. But for exchanging materials with the surrounding, the cell also uses two famous processes. They are called as diffusion and osmosis. Diffusion means the movement of molecule from a region of high concentration to a region of lower concentration. The common examples of diffusion that we can see in our everyday life is once we spray a perfume in one corner of the room, the fragrance just migrates or travels to the other part of the room also. That's simple diffusion that is taking place. Or when we dip a tea bag in a cup of hot water, the flavors from the tea bag will diffuse it into the water. Cell also follows osmosis and diffusion, that is movement of substances from a region of higher concentration to a region of lower concentration. Now, osmosis is a type of diffusion, but here it is the net movement of water molecules from a region of higher potential to a region of lower potential. So in this figure you can see I have created a setup for diffusion and osmosis experiments. If you see, in case of diffusion, you take a high solute concentration, put it into a solvent, and within a short period of time the solute just migrates to different corners of the solvent and attains a balance, a equilibrium. In case of an osmosis, if you are trying to separate two compartments of a beaker with a semi-permeable membrane, with one half having low solute concentration and the other half having high solute concentration, after some time the equilibrium will be reached and both sides will attain a balance. Now what according to you is a solution? Solute plus solvent makes a solution. There are three types of solutions we can say. We have the first figure, that is isotonic solution, where the concentration of the solute and the solvent is almost the same. Whereas in case of hypertonic solution, the solute is more as compared to the solvent, a concentrated solution. Whereas in case of hypotonic solution, the solute is less and the solvent is more. We can call it as a dilute solution. Animal cell and plant cell behave differently when you keep them in these three types of solution. So on the slide over here, we have cell A, B, C. One side is the red blood cell, that is animal cell, and the other side is a plant cell. When you keep them in different solutions, each will show a different kind of a behavior. For example, if you're keeping the cell in plant and animal cell in isotonic solution, they will attain a balance in such a way that the exchange of materials within its environment will be the same and the cells will remain as it is. Whereas if you keep the cell in a hypertonic solution, a concentrated solution, in case of an animal cell, the animal cell will lose a lot of water molecule from its body and the cell will shrink. In case of a plant cell, we have a cell wall. So the cell wall will not shrink, but the plasma membrane as well as the vacuole starts to shrink and comes to the center of the cell. If you keep a plant cell in a hypertonic solution for a long time. But if you keep an animal and a plant cell in a hypotonic or a dilute solution, what happens? The animal cell will first keep on absorbing the solvent, but after some time the plasma membrane of the RBC cell must be stretched to such a huge extent that the cell cannot withstand the pressure and it just bursts. In case of a plant cell, the vacuole and the cytoplasm and everything gets so full of the solvent that they start to exert a pressure towards the outside. But at the outside of the cell you have the cell wall which exerts an equal pressure towards the inside. Thus, the plant cell maintains its structure. Such a cell is called as a turgid cell. The plasma membrane also shows another phenomena that's called as endocytosis, wherein a cell can take in a substance by surrounding it with its cell membrane. You must have studied about how amoeba captures its food using pseudopodia and all. Well, that's the plasma membrane of the amoeba which is so flexible enough and allowing amoeba to do that. We also need to talk about the cell wall. Cell wall are most commonly found in plants and they give a structural strength to the plant body. Cell walls are also present in fungus, fungi and bacteria. Of course, the chemical structure is different. Let's come to the nucleus. Every cell will have a nucleus and the nucleus has four major parts that you should know. The outermost covering of the nucleus is called as a nuclear membrane, which is double layered, and there are certain gaps or holes in the nuclear membrane which is called as a nuclear pore. It is through this the nucleus exchanges materials with the cytoplasm. Inside the compartment of the nucleus you have the nucleoplasm, and in the nucleoplasm are suspended thread-like structures called as the chromatin network. We also have a nucleolus inside a nucleus. What is this chromatin network? Chromatin network is nothing but the DNA and proteins. We cannot see them regularly, but at the time of cell division, the entire chromatin network gets condensed to form a proper chromosome structure. The nucleus of a cell has very important roles to perform. It plays a central role in reproduction. It helps to determine the way the cell is supposed to develop and grow and mature. The nucleus also will direct the various chemical activities inside the cell. There are two types of cell based on whether the genetic material is covered by a nuclear membrane or not. And these two types of cells are called as the prokaryotic cell and the eukaryotic cell. A prokaryotic cell will lack a nucleus and other membrane bound structure, whereas a eukaryotic cell will have a nucleus and membrane bound structures. Prokaryotic cells have a small ribosomes, whereas eukaryotic cells have large ribosomes. In case of a prokaryote, the DNA is not organized into chromosomes, whereas in case of eukaryotes, the DNA is organized into chromosomes. In case of prokaryotes, the flagella are not made up of microtubules, whereas in eukaryotes, the flagella are made up of microtubules. The cell walls of the prokaryotes and the eukaryotes also are different. So here we have a figure of a prokaryotic and a eukaryotic cell. You have a figure of them in your textbook also. Please go through it. There are n number of cell organelles inside the cell, and together they perform all the activities and keep the cell working. So on the list you have the various cell organelles. You have the cytoplasmic membrane or the cell membrane. Then comes the endoplasmic reticulum, ribosomes, nucleus, nucleolus, nuclear membrane, Golgi apparatus, cytoplasm, mitochondria, chloroplast. Each one of them has a particular role to perform in the plant body. Endoplasmic reticulum. These are network-like structures that are in continuation with the nuclear pore, and it is through this that many materials are passed around in the cell. You can see in some areas the endoplasmic reticulum has small dot-like structures. Those are nothing but ribosomes, which gives them a rough appearance, hence the name rough endoplasmic reticulum. Whereas in the smooth endoplasmic reticulum there are no bumps or ribosomes, hence they have a very smooth appearance. Endoplasmic reticulum is commonly called as ER and is also called as the highway of the cell. Okay, the rough endoplasmic reticulum, because they have ribosomes attached to them, they are responsible for protein synthesis. Whereas the smooth endoplasmic reticulum usually is just goes on creating lipids. The Golgi apparatus is a structure which resembles a stack of plates and is lying very close to the endoplasmic reticulum. It accepts materials from the endoplasmic reticulum, stores, modifies them and packages them into protein. It is by small vesicles that the molecules are transported into and out of the Golgi apparatus. Lysosomes are the scavengers of the cell, basically related with disposal of waste in the cell, but they also contain some very powerful enzymes that has the capacity to destroy entire cells. The mitochondria is called as the powerhouse of the cell. It is here that cellular respiration takes place and ATP is generated. It is also a double membrane bound structure and has its own DNA. Plastids are called as the kitchen of the cell, basically the plant cell. Plastids are found only in plant cell, and you have three types of plastids. You have chromoplast, chloroplast and leucoplast, based on the pigment that they possess. In case of leucoplast, they don't have any pigment, but They are responsible for the storage of starch. Vacuoles are large storage structures that are very prominently seen in plant cell. Doesn't mean they are absent in animal cell. They are there, but they are tiny in structure in the case of an animal cell. Okay. Now, the vacuole plays as a storage organ for water, food, enzymes, pigments, etc. Now, let's have a new concept that's called as cell division. Now, cells in our body divide. Why do they divide? They want to grow, they want to replace the old cells, and sometimes cell also divides for the production of gametes that will later on take part in reproduction. There are two types of cell division, that is mitosis and meiosis. The basic difference between them is, in case of mitosis, the parent cell is dividing into two daughter cells, whereas in case of meiosis, the parent cell gets divided into 4 daughter cells. In case of mitosis, the number of chromosomes in the parent cell and that of the daughter cell is the same, whereas in case of meiosis, the number of chromosomes in the daughter cells is exactly half the number that is found in the parent cell. Mitosis helps us to grow and repair our body, whereas meiosis are solely responsible for the production of respective gametes.
So my dear students, we have come to the end of our chapter and let's recollect some of the topics that we covered in this chapter. We started our lesson with the definition of cell. From there, we studied how the cell was discovered, the various components, how they were discovered in the entire process. We also studied about cell theory. We studied about the various components of cell, their organelles, their respective functions. We also tried to differentiate the way an animal cell and a plant cell will behave when we put them into different solutions. We also tried to distinguish between a prokaryotic and a eukaryotic cell. Students, at the end of this chapter, I would like to request you to appreciate this tiny bit of life called a cell, which makes every living thing live on this earth. Thank you.